Svenn-Erik Mamelund is founder and head of Center for Pandemics in Society at Oslo Metropolitan University. He looks at historical pandemics through a social lens and how socioeconomic factors define how pandemics affect us today. Welcome to the Letten seminar, Professor Mamelund. We're all ears.
Thank you. Thank you so much for that nice introduction. I believe that serendipity or curiosity, as well as stamina or endurance, are two key components in doing excellent research that also has societal impact. So in my talk, I will briefly present my own serendipity moment in research, and then move on to two examples from my own career to look at the impact of endurance on my own research and also of others. In the summer of 1995, 28 years ago, I was a research assistant in Statistics Norway. And I was employed there to do a report on the mortality development in Norway over the 150 last years. And the first graph I made when I was in this RA position, this research assistant position, was this graph that you see on the left-hand side here. This is showing the number of deaths in Norway from 1893 to 1992. And what struck me, obviously, was this peak in 1918, with 45,000 people dying in this country, in Norway, with 30,000 dying just a few years before, and also some years after the pandemic. And I was like, what could potentially kill 15,000 people in Norway? It's 0.6% of the population of Norway at that time. And I was asking my co-author, what could potentially take so many lives? And at first I said, could it be the war, the First World War? And then we quickly agreed that, well, Norway was neutral, so it couldn't be due to the war. And then he said there was a flu going on at that time. And I was like, flu? Isn't that just a disease that kills some elderly and some frail people each year? It can't kill an excess of 15,000 people in 1 year. That can't be true. And then he told me that, well, this was different. This was a pandemic. It was a new virus with that few had immunity to fight this disease. It was called Spanish flu. It killed young adults, people in the ages 20 to 40. And globally, it killed up to 100 million people. So up to 5% of the population at that time actually died from the Spanish flu. I got totally in love with this topic and started to read the literature. There were very little literature on the Spanish flu in Norway, so I had to read all the international literature. And I asked my co-author, Jens Christian Borgan, whether we can have a chapter in this report on the Spanish flu. And then he told me that, Svenn-Erik, this was supposed to be a report on the general period and cohort mortality patterns in Norway, and not a feast on Spanish flu, so to speak. But I insisted on having a chapter on the Spanish flu in the report, and in the end, Jens Christian Borgan agreed that we could have a report or a chapter on the Spanish flu, and it triggered and was important for all of my career, I would say. If Jens Christian Borgan, as the senior scholar at that time, had sort of stifled my curiosity at that time, at sight, so to speak, 28 years ago, I don't think I would have done my master's thesis on the flu, my PhD, my postdoc, and later I got funding to do research on pandemics, and also to lead PANSOC, the Center for Research on Pandemics in Society. So this was just so important to me. So I guess my message to senior scholars and to senior faculty is that let junior scholars and junior faculty follow their own dreams and not yours, and their own curiosities, if if possible. I'm now moving on to a table showing the risk factors for disease outcomes during seasonal flu, Spanish flu, and COVID-19. And I'm showing this to remind the audience that risk factors are not only medical ones, like high age or non-communicable diseases or pregnancy and so on. Low socioeconomic status, race and ethnicity, and also indigenous status is actually established risk factors in the literature, and they are often, not always, but often important above and beyond controlling for underlying comorbidities and so on. So it's important to get the full picture of disease burden. You need to look at both biological and ethnic and social risk factors. So elderly is normally at risk for flu, but not so in 1918. So you see in this graph, that there's this classical U-shape of mortality for seasonal flu, but in 1918, it was a W-shaped pattern with the increased mortality for young adults. We actually know too little about why young adults were so hammered in 1918, but for now, let's focus on why the elderly seemed to escape in 1918. And this could be due to the fact that there were H1 type viruses circulating before 1889 that the elderly was exposed to and could develop some type of immunity to fight the Spanish flu in 1918, and hence have some immunity to fight off that flu. And here's real data for Norway. This is P&I mortality data. This is pneumonia and influenza mortality in Norway. And the upper solid graph is showing 1918, and the one at the bottom is showing 1917 as an example of a baseline year. And what you clearly see is that mortality is driven by the young adults. And you can see this even more clearly here. In this graph, this is relative to total mortality for all causes of death, both genders or sexes combined, 1-year age groups, and there's a peak in mortality among young adults, and there's a decline in mortality over age, and even a lower mortality than expected for the elderly, which hints to this idea that there must have been some type of immunity among the elderly. And if you guys remember the swine flu pandemic, the same thing happened in 2009. This was an H1N1 virus, just as the one that caused the Spanish flu. And the elderly actually fared better than young adults, also in 2009. And people that were born close to 1918 and that was exposed in 1918 or in the 1920s, they had more antibodies in their blood, and hence immunity to fight the pandemic we had even in 2009. And this also shows that pandemics are connected over time. And this is an important message for me as a social history historian. And so if elderly in urban societies had rest immunity, like I showed you for Norway, I could have showed other figures for Sweden and for other industrialized societies and countries with the same patterns of mortality. But a paper came out in 2007 showing or suggesting that maybe there would be less rest immunity among elderly in isolated places on the planet. But at that time, in 2007, nobody had collected empirical data to look at this issue. And so I started to collect data in 2007 for isolated places on the planet. And this is what I found. A staggering mortality of 27%. So this is 27% of the population in Labrador dying. On Western Samoa, 24% died. Alaska, on average, these are areas that we know that got the disease, had 8% mortality. And Enara, this is a Sápmi area in Finland, had 10% mortality. Arjeplog, Sweden, another isolated Sápmi village. This is Sweden. And at the bottom, Karasjok in Norway, 2% mortality. And you also see sort of an ethnographic or qualitative image or sort of evidence of those survivors that you could expect from this flu in isolated communities. It would be the ones between the ages of 5 to 14. And you see this even more clearly here for Brevik, Alaska, where 90% died. 80 people lived in this village before the flu, and 72 died. So 90% mortality. And the only survivors are a handful of kids in the age of 5 to 14, as you see on this graph. So what's in the black box? I did a study 20 years ago for Norway showing that there were higher mortality in areas of Norway with a higher number of Sami people. So even after controlling for a number of confounders, there was a higher mortality in these areas. So I suggested that geographic isolation could potentially explain why they had a higher mortality. So hence, less exposure to H1-like viruses in the past, and then less immunity to fight off the Spanish flu in 1918. But that time, I only had like aggregate level, municipality level data. So you really need individual level data to look at the hypothesis of geographic isolation as a key to explain these differences. So moving 20 years on to this year, and this paper that we just published in International Journal of Circumpolar Health is part of the CAST project we just did. The CAST is Center for Advanced Study. We did a project on indigenous peoples and pandemics last year, and this is one of the results. And we were now the first, to do look at individual level data to look at this hypothesis, and as one reason for higher overall indigenous mortality. And so the context, obviously, for those that don't know the context, we looked at Kautokeino and Karasjok. It's pretty much a Sami-dominated area. It's, two of these parishes that we look at are among the largest in 1918 and also today. Very small populations, only 1,000 living in each of these places, and low densities and so on. No roads connecting these places to the coast and so on. So it's really, really isolated places in a Norwegian context. So we look at burial registers and censuses, and we estimated all-cause mortality, excess mortality for 3 age groups. And I'm going to show you the results for the excess mortality in a minute, but I'll just want you to look at the map to the left, showing the diffusion of the disease in Norway, showing that it started in Oslo, Christiania, and it moved on by people traveling with coastal steamers. It came to Finnmark, to the uppermost county, northernmost county of Norway, where Karasjok and Kautokeino is situated. It spread to the coastal areas, but it didn't spread inland in those 2 areas that we look at. So we have 2 peaks in Karasjok. One is in October and November 1918, and a second one in February 1920. And Kautokeino just had a major outbreak in January 1919. And here we also see that all age groups actually had excess mortality as expected in both areas and also over time. So we saw no real summer wave in these isolated areas. There were delayed disease onset. There was a February 1920 wave in Karasjok and excess mortality for all groups. And all of this is consistent with this idea about remoteness being key to explain these differences. So all age groups had excess mortality, and not only young adults as in urban and non-isolated societies. And this hints to this idea about geography being very, very important. And finally, I just wanted to show this. This is Johan Hultin. He is in this mass grave. You remember this place in Brevik where 80 people lived before the pandemic? 72 died, and they made a mass grave in the permafrost. So 72 people were buried in the same mass grave. So Johan Hultin here, he's a Swedish virologist. He stands in the middle here. What they wanted to do, talking about curiosity and endurance and so on, I mean, in 1918, the virus wasn't isolated, and the cause of flu was discovered in the early 1930s. So obviously what they wanted to do here was to sort of get a tissue of a lung from a person dying from flu, go back to the labs and try to sort of tease the virus out. But they were unsuccessful in the 1950s. 46 years later, however, Jeffrey Thunberger, a colleague of mine, started to look at lung samples taken from US soldiers. But as you can see here, those lung samples are hard to work with, they are small and so on. And Johan Hultin, 46 years later, he read about this in Science, and that Jeffrey Thunberger was struggling to get something out of these lung samples. So he gave him a call and said, Jeffrey, do you want a lung? I have a lung taken from a woman that died from the Spanish flu up in Brevik, Alaska. So I know you need this in order to do your work. And obviously he said yes, and they met, and 10 years after they met, they mapped the full gene sequence of this virus, and also the lethality of the virus was confirmed in 2007. But there's a ton of things we don't know about this virus yet, so we need more research. We always say we need more research, we need more money, and we need to do more research where social scientists also collaborate with people, lab people, wet lab people, and biologists. Thank you so much for your attention.
Thank you, Svenn-Erik. Could I just ask you one question, please? I just got so curious because these numbers, we have been talking about numbers. We need good numbers earlier today. And we have mortality rates and good numbers from 1918. Really?
Yes, you would be quite surprised actually that people actually did surveys back then, which are really, really well done and carried out. There were data from insurance companies that are good. There are census data, there are routine notification data, and some of these data are actually as good as the data that we can sort of bring out today. We have seen it during COVID, that we actually have a hard time sort of estimating R naught and disease burden and so on. We have a lot of troubles also today.